This video is going to be about two topics. One of them is going to be ATP, which you will remember, I'm sure, from some previous course, and metabolic pathways. So it's going to be important to understand what a metabolic pathway is and to understand the deal with ATP. So let's just get into it and go over those topics. So ATP is a big deal in most known types of cells. It is involved in two major reactions. One of them is the one where an enzyme will break down a phosphate-phosphate bond and release one of the phosphates. So adenosine triphosphate has 3 phosphate groups. So it'll release one of them, and that releases enough energy that the enzyme can take that energy and do something with it. So if an enzyme breaks the phosphate-phosphate bond and converts ATP to ADP, that enzyme gets energy, and that's often used in chemical reactions in a cell to do reactions that require energy. So that's the most common source of energy for enzymes in cells. The other kind of reaction it goes through is the one where it is made from ADP. So ADP can be converted back to ATP if energy is supplied. So think about the energy that an enzyme can take from this reaction. It takes slightly more energy to put that phosphate back onto an ADP and remake it. But the thing is that source of energy, that ATP, is so useful to enzymes. So many different parts of so many cells have evolved to use this as a source of energy that every kind of cell is working all the time to convert ADP back into ATP. So to constantly have a store or stockpile of ATP. And cells go through this very quickly. They'll use up all their ATP and remake it very quickly. So these are the two reactions. Hydrolysis is the one where ATP combines with a water molecule and breaks this bond, and the water molecule kind of takes its place. And that's where an enzyme will take that energy and use it. So the energy is released from this, and the enzyme, and many different enzymes, can take that energy and use it. So then what you're left with is adenosine diphosphate and 1 phosphate group. I should back up. This is a nucleotide triphosphate. Adenosine is the A in ACTG. So this would technically be the A from RNA, because this is a ribonucleic acid. So this is the nucleotide base, or the nitrogenous base, adenine. And when you combine that with the sugar and with the 3 phosphates, you call that adenosine triphosphate. So this is whatever, it's what it is. This is the form with just 2 phosphates, so adenosine diphosphate. Now, if a cell wants to make this back into ATP, it has to put this phosphate back on and rebuild that bond, and that requires energy, so we call that synthesis, making ATP, or generating ATP. It's always a little bit awkward to talk about this because this ATP is almost made. It's ADP is everything except this phosphate. So all we're really doing is rephosphorylating, or adding a phosphate to, but we typically talk about it as making ATP or generating ATP. So that's ATP. That's what you need to know. If that was review, good. If not, do what you must to understand it. And we get into metabolic pathways. So a metabolic pathway is a sequence of reactions where the product of one reaction becomes a reactant of the next reaction. And cells use these sequences of reactions whenever it makes sense to do the same sequence of reactions over and over again. Cells will have many interconnected pathways that they can use for different reasons. But in every case, enzymes are going to convert an initial molecule, a substrate, into a bunch of other molecules, and eventually what we call a product molecule. So one example is the metabolic pathway glycolysis, which starts with glucose and converts it to pyruvic acid. And that is a big part of energy metabolism in cells. So this slide has a lot on it, but it's a made-up pathway to kind of illustrate what goes on in a pathway. So if you look at this molecule, you may or may not recognize this as a five carbon ring with an oxygen. So like, well, certain sugars. This is a sugar of some kind. So this is a real molecule. It's just I don't know what it's called and no one cares. So this would be the initial substrate in the pathway. And after the first reaction, it's different. So what this reaction has done is change this alcohol group, this OH, into a phosphate. And water would have been released. So this would have been converted to water and replaced with this phosphate. So we go from this molecule to this molecule, and the only difference is this phosphate. And then the second reaction does the same thing in this position. So now this molecule has 2 phosphates. And then the third reaction does the same thing at this position. So now this molecule has 3 phosphates, and this would be the final product of this made-up pathway. So some things to note. An enzyme will catalyze these reactions. Enzymes typically catalyze these reactions, and enzymes are very specific. So usually the enzyme that will catalyze this reaction could not also catalyze this reaction. That's because it has to perfectly fit this molecule. And once you add a phosphate to it, it's very different. The same, nothing that fits this perfectly would also fit this perfectly. So chances are there'd be 3 different enzymes that would catalyze these 3 different reactions. Even though each reaction is sort of doing the same thing, they are distinct reactions, and that's what it is. So again, enzymes are catalysts, and if none of that was familiar to you, catalysts make reactions happen faster. That's all they do, and they're not changed in this process. So a catalyst can be reused again and again and again to keep making a reaction happen very quickly. Enzymes are just proteins that act as catalysts, but they are really specific to the substrate molecules they work with. And in cells, having an enzyme for a specific reaction makes that reaction happen very quickly, even though normally the reactions that happen in cells would never happen without a catalyst, or it would take them a year to happen instead of a microsecond. So catalysts are the difference really between reactions not happening and a reaction happening right now. So cells can use that to basically turn on chemical reactions, to make chemical reactions happen. And so every pathway is controlled by whether or not the enzymes exist. So when a cell wants a pathway to go, it makes all the enzymes, it makes all those proteins. And this is what lets cells control their internal chemistry. If you think back to redox, you'll remember that some molecules are good at donating electrons and some are good at accepting electrons, and others are in the middle, and those can work as carriers. So there are 2 electron carriers we're going to want to pay attention to in the coming slides. One of them is NADH. Now this molecule comes in 2 forms. One is NAD+, one is NADH. NADH is full. It is carrying 2 electrons that it can then donate to some other molecule. NAD+ is empty. It can accept 2 electrons and then become NADH. So if NAD+ accepts 2 electrons, it will become NADH. And if NADH gives away 2 electrons, it will become NAD+. So I just think of this as the full form, and this is the empty form. And then there's a similar thing with FAD. This has 3 forms. So FAD, if it accepts 1 electron, it becomes FADH. And if it accepts 2 electrons instead, it becomes FADH2. So these are electron carriers, and it's important to recognize when they are empty and when they are full, because that can tell you what happened in a chemical reaction. if an electron carrier goes from empty to full as part of a chemical reaction, it took electrons from something. And that's going to happen a lot in cellular energetics. So that was ATP and biochemical pathways. That was a whole lot of random jumbled stuff thrown at you, and we will use it all in the next video or 2 or 3. So I will see you there.